Permission to record and share this lecture.
Everybody, we'll get started. So, as most of you know, my name is Cinnamon, and I have the great pleasure of introducing our speaker today. You guys might not know her, but I think you're going to start to recognize her wandering the halls here very soon. So this is Susanne, I'm going to say your name wrong again, Brander. And she is currently over on main campus, and she works with the environmental toxicology and chemistry there. And she's going to actually start doing some work here with us in one of the cold rooms off of Lab 27. So that's why you're going to see her pretty soon. Her master's work was done at Johns Hopkins, and then she did a lot of work at UC Davis with her PhD and several postdocs. She also did some postdoc work at UNCW, where she also worked before she came out here to Oregon. So we're excited to have her here on main campus, and we're even more excited to have her join our Hatfield community. So before I hand it off to her, I also wanted to remind you guys that next week's seminar is Gail Hansen. So if you want to learn a little bit more about the work that she's been doing with the tsunami debris and the algae that's come on that, please join us again next week. Other than that,
Thank you, Cinnamon. All right. Great to see everyone here today. I'm excited to be out here on a beautiful sunny day. This morning I opened my email and it said school was canceled and there was going to be this giant snowstorm, and then I got, wow, this is gorgeous. So anyway, great to see everyone today. Like Cinnamon said, I am an environmental toxicologist. I consider myself to be an eco-toxicologist. I mainly focus on marine and estuarine systems. I think a lot about fish, and today's talk is going to mainly focus on studies in fish. But I've also done work in crustaceans. I do some work in shellfish as well. So I'll talk a little bit about other examples towards the end of the talk. So the title of my talk is I tried to make it pretty broad. So from the benchtop to the beach, assessing responses to environmental stressors in marine and estuarine organisms. So when I talk about environmental stressors, I'm not just talking about chemicals, also talking about things like global climate change. I'm sure a lot of you have heard about the problem of marine plastics, so also thinking about that issue as well. And thinking about things from the smaller scale, so at the molecular level, what's happening with gene expression, for example, to try to think about that and take that up to the population level where possible as well, with lots of collaborations. So I wanted to start off the talk by thanking and acknowledging all of my collaborators and students. It wouldn't be possible to have done any of this without. Like Cinnamon said, before coming to OSU, I was faculty at the University of North Carolina, Wilmington, on the other side of the country for about 3.5 years. So a lot of this work was done there. So to introduce things, my group really investigates the effect of both toxicant, so pollutant, and general stressor exposure in a variety of organisms. So we think about gene expression, protein expression. Sorry, some of this is a little bit small. Think about multiple stressors. So what happens if you're exposed to a higher than normal temperature and a pollutant simultaneously? So it's trying to take it out to the real world. Developing ecologically relevant model species, and that'll really be the focus of my talk today. I'll talk about 3 different fish species that we're trying to develop as more relevant models specific to the problem at hand. Thinking about multiple biological scales, so how do you take something that's happening at a small scale and use it to predict what might be happening at a scale that's relevant to risk assessment? So behavioral effects, and also just thinking about the bigger picture and how it all comes together. Right? So research goals in general are really to choose the right model system. So instead of having one model species that my lab works with, we tend to work with lots of different models, fish and sometimes invertebrates as well, aiming for environmental relevance. So not just thinking about what's happening to the fish in that beaker in the lab, but thinking about what's happening to it in the estuary or along the coast, and trying to push the boundaries a bit of what risk assessments should consider, right? Thinking about what, if it's assessing risk, is it really evaluating the risk in a realistic manner? Right, so the stressors that my group thinks about are pretty broad. I've done a lot of work with endocrine disrupting compounds. And so these are chemicals that you may have heard about in the news or in your own research, but chemicals that interfere with hormone function. So something that people might have heard about is birth control pills. So birth control pills get into the environment through treated wastewater effluent because wastewater plants don't remove it. And then fish get exposed to that. And it turns out that fish and other aquatic organisms respond similarly to that as we do. It acts as an estrogen, right? It interferes with reproduction, interferes with growth. Lots of different categories of endocrine disruptors, everything from drugs, that doesn't work, I have to remember that, to things like pesticides and personal care products, right? Also been thinking about climate change. And so I'll talk about a study that a PhD student of mine who has recently arrived at OSU to finish her degree has completed, where she looked at combined exposure to a warmer temperature and a couple of pesticides or pharmaceuticals that fish are routinely exposed to. We think about this in terms of what's happening in an estuary for that particular model. And then we've also started to think about microplastics, and not just the ingestion, the direct ingestion of microplastics by fish, but how are they accumulating those plastics from their prey? And so I'll talk a little bit about that as well, and in the context of species, fish species that are consumed by us, right? So one of the main models that my group works with is Menidia beryllina, which is the inland silverside. So this is a fish that's very important in estuarine food webs. It's kind of smack in the middle of the food chain, and so it's eaten, and it gets eaten, and it eats lots of lower trophic level organisms. It's found throughout coastal North America, so it turns out it's not found off the coast of Oregon, but it is found in California. It's found in the East Coast and the Gulf. It has a short generation time, and we've recently sequenced its genome, right? It readily spawns in the laboratory, and actually the cold room that Cinnamon was referring to is where that is hopefully going to be happening in a few months, so we'll have a broodstock population there. And they're interesting in terms of thinking about global climate change because they exhibit something called temperature-dependent sex determination. So basically what this means is that although they have genetic sex determination, their sex is also determined phenotypically by the temperature at which eggs are incubated at and those early larval stages experience. And so if those early stage organisms are exposed to colder temperatures, they tend to be more likely to become female. They have a female biased sex ratio. If they're exposed to warmer temperatures, they tend to have a male biased sex ratio. The reason for that is that females are able to produce more eggs, are more fecund if they're larger. And so having that sort of head start early in the season when it's colder gives them more time to grow. And so we'll come back to this later when I talk a bit about the effects of warmer temperatures, concurrent exposure with endocrine disrupting compounds. And a lot of studies, both in my lab and in other labs, have shown that Menidia are sensitive to endocrine disrupting compounds. So things like altered sex ratios, altered gene expression, which I'll give some examples of. Right. So the first study I'm going to talk about kind of sets up the next two that are more recent. And so for a number of years, I've been developing this model as a model to predict the effects of endocrine disrupting compounds. And again, these are chemicals that can interfere with reproduction, which is kind of the first thing people don't really think of, but they also interfere with growth. They interfere with immune response, right? They interfere with metabolic rate. So the endocrine system controls many biological functions beyond reproduction. And so we're trying to think about this with a systems biology approach. So trying to assess what's happening at the lower level and using that to inform higher level responses. And so we've done studies where we've started by sequencing the transcriptome, and basically by that I mean looking at all of the mRNA that is being expressed at a particular time, all of the genes that are being expressed in that organism. And then looking at what a particular model EDC influences. And so we know that they're sensitive to endocrine disrupting compounds, because if you expose them to a very low level of a model EDC, so in this case it was half a nanogram per liter, so you're thinking half a drop of this chemical in an Olympic-sized swimming pool, if you're trying to think of what the relative amount is, you're seeing changes in the expression of genes that are related to all these different functions, so growth, reproduction, immune response, even things like oncogenesis, so tendency to develop tumors, for example. And so what we've seen in this species is that when you see changes in those types of genes, and genes that have those functions, that can also be connected to changes in reproductive capability. And so in this case, we saw that when they were exposed to that model endocrine disruptor, it caused them to produce less of a protein that's used to make eggs. So I don't know, how many of you have heard of the protein choriogenin? It's okay if you haven't, but it's the protein that makes up the coat of the egg of a fish. And so this is true across most fish species. Choriogenin is the egg coat protein. Vitellogenin, which you may have heard of, that's been used as a biomarker for endocrine disruptor exposure, is the egg yolk. Well, you expose them to increasing levels of this chemical, they make less egg coat protein, and then that corresponds to a reduction in egg production, which makes sense. If you're making less of the protein that comprises the egg, you're going to make fewer eggs in general, right? And then you can scale that up, and I won't get into the model in detail here, but you can use a density-dependent, age-structured model that uses both the choriogenin levels, so the egg protein levels, and results of spawning assays, so how many eggs are they laying. You can use those to inform the model to predict what the population size is going to do over a number of decades. And so in this case, you can see that we showed a 30% reduction in population size following exposure to this model endocrine disruptor that acts as an estrogen that interferes with estrogenic signaling. So that kind of sums up why we've gone with this particular fish as a model to do other studies that build upon these findings. And so what I'm going to talk about today are two additional studies with the inland silverside. So one looked at combined effects of increased temperature and endocrine disruptors. The other looked at effects of early life exposure just to a parental generation, and then took that out to additional generations to look at effects that carried over. And then I'll talk a bit about the trophic transfer of microplastics, so the transfer of microplastics from prey to predator. And lastly, I'll finish up talking about a recently completed experiment looking at a deep sea model to assess the effects of oil spills. Right? So to start off, after we established that silversides were highly sensitive to endocrine disruptors, the next question was, well, what happens to their offspring, right? There has been a lot of published literature in the past 10 or so years, a lot of it on mammalian species, but we're starting to get more information on zebrafish, for example, that stressors, and we're not just talking about pollutants, we're talking about situational stress. There have been some studies that in humans, for example, that have shown that the grandchildren of people who were held in Nazi concentration camps have a higher occurrence of certain stress-related disorders, for example. They've shown this by looking, there's some crazy studies, I don't know if anyone listens to Radiolab, but where they've looked at the records from churches, I think in Finland or Sweden, and they're able to trace back the effects of famine. People who were exposed to famine back in like 1910 have great-grandchildren who have metabolic disorders, certain rates of metabolic disorders. So they're seeing this in humans, they're seeing this in mice, they're seeing this in plants, and they're seeing it in fish. And so we wanted to know what happens if you have potentially this ability to have these transgenerational effects, phenotypes passed down. And you're also thinking of it in the context of climate change, so increased temperature. And we already know that these fish are really sensitive to low levels of endocrine disruptors that you actually will see in the wild. So what happens? And so my PhD student undertook a study where she exposed silversides to both a couple of endocrine disruptors and warming temperatures. So this, we thought would be important for understanding effects on chronically exposed wild populations who aren't just exposed for a few days, but over a lifetime. It allowed for the testing of effects of parental exposure on offspring, and it was able to determine if effects were going to be additive across generations. I don't know that we could address whether they adapt because we only went out to the F2, but something that's a goal for future studies. And again, we've already established that they're sensitive. So what she did was she acclimated silversides to 2 different temperatures and exposed them to environmentally relevant levels of 2 endocrine disruptors. One is a pesticide, bifenthrin. Ethinylestradiol is what you're going to find in most birth control formulations, right? So pretty commonly detected compounds. And what she did is she exposed the parental generation, and then she continued to expose their offspring through early life, and then she reared them up in clean water until adulthood and spawned the F2 generation, right? So this was the exposure, and then following that 21 days post-hatch in that second generation, they were reared in clean water, right? And so this is what we found. We found that sex ratios were altered. And remember we talked about earlier how silversides have something called temperature-sensitive sex determination. So if you look at the controls, it's as expected, right? At the colder temperature, you have a higher proportion female. At the warmer temperature, you have a lower proportion female. You get more males, as expected, right? If you expose them to bifenthrin, this difference is no longer as significant, right? So you're seeing that you're getting more females at the warmer temperature. If you expose them concurrently to ethinylestradiol, which is basically designed to act as an estrogen, it's not, bifenthrin is more of kind of an accidental estrogen. We didn't realize it was going to act that way. You see a reversal of that sex ratio. So instead of getting more females at the colder temperature, you're actually getting more females at the warmer temperature. So what does this mean adaptively? We're still thinking about how to input this information into a demographic model that would give us some predictions on what this would mean at the population level. So we also saw deformities, and these deformities appear to be influenced in some cases by the temperature at which the organisms were exposed. And so although it wasn't significant, we had a trend towards having more deformities in the pesticide-exposed fish at the warmer temperature. And then in the F2 larvae, so these were the larvae that were not directly exposed, we saw more defects, and these were caudal fin deformities, where they were basically missing pieces of their caudal fin. We saw more of those deformities at the warmer temperature, particularly with ethinylestradiol. Right. We also saw changes in gene expression, and so I won't go into a lot of details on the particular function of these genes, but basically we tried to hone in on genes that regulate reproduction, that regulate the fish's ability to grow, right, to respond to pathogens. And so what we saw here was that this gene is important for steroidogenesis, so the production of androgens, the production of estrogens, in which fish are very similar to humans. We saw that this gene was downregulated. Less of it was expressed in both the F1 and F2 larvae that were exposed to that pharmaceutical. And then if you looked at this other gene, which binds to estrogen, so it's a cellular receptor that binds to estrogen, you also see altered expression. Although in this case, it didn't carry over between the 2 generations. We saw differential expression, although in both cases, it differed from control organisms. So lots of work left to do in that area. But to summarize, both warmer temperatures and exposure to endocrine disruptors resulted in reduced larval production, right? It seemed like it was a bit worse in the F2 generation, which to us was surprising because they weren't directly exposed. Sex ratios were influenced by combined exposure to EDC and temperature. We saw deformities, and in some cases that seemed to be influenced by temperature, more so in some cases than others. So stronger influence in the F2. And then some effects of gene expression seem to be passed on to that F2 generation. And some of these could lead to things like altered levels of hormones that are important for reproduction and growth, or even things like oocyte development, which might explain what we were seeing with the reduced fecundity in fish exposed to some of these chemicals in the first study I talked about. So we decided to build a bit on this study, and actually right in the middle of doing that work, which took about 18 months or so to get those fish through all those different generations, we got funding to do another multi-generational study. And this time we decided to try to take it a little bit further and get more at the mechanisms underlying these transgenerational effects. We said, okay, what happens if you just expose the parents? We're not going to expose the parents in the F1. We're just going to expose the parents. And we're only going to expose them for the first 21 days of life. Because some of these mammalian studies and studies that have been coming out in other more established models indicated that really that period in early life was the most sensitive period for having some of these effects that tend to carry over. And so basically there was a gap in understanding these effects that are often termed epigenetic effects in fishes. And initial studies suggested that they might be severe. And so what we did was we isolated the exposure to just that early life period. So questions were, what are the effects on reproduction? What are the effects on immune response? And what is the potential for fish to pass on these effects to the F1 and potentially F2 generation? And so if you think about it, the experiment we set up here, we were directly exposing the parents, but just in early life. And then that F1 generation, you can think of it as being unexposed, but they are being exposed if you think about the fact that they do exist as primordial, basically pre-sperm and pre-egg cells in their parents, even when their parents are at a very early life stage and aren't reproductive yet, aren't reproductively mature. So in the F2 generation, when you're thinking about a fish, is the generation that hasn't been exposed at all. So we really wanted to isolate the exposure to the parental generation so we could get at this unexposed generation at the F2. Right? And so again, we were trying to look at these effects across scales. And so we looked at a couple of different chemicals. Some of them were the same ones we've looked at before. So ethinyl estradiol and bifenthrin, which is a pesticide used on mosquitoes. Levonorgestrel is also used in birth control pills. It's a synthetic progestin. And then trenbolone is an androgen, and it's used to promote cattle growth. And so it's been found at elevated concentrations in runoff from feedlots, for example. So F0 and F1 were evaluated both as juveniles and as adults. And the F2, we didn't have time to grow them out until adulthood, and so we evaluated them also at 21 days post-hatch. Right? So a couple of preliminary results from this work. We're still actually a bunch of fish just arrived in Oregon a couple of weeks ago, and they're in my freezer. So we'll be getting to the F2 generation shortly. But as a primer, we have seen some effects in the parental and F1 generation. And so, like we've seen with other studies, we're seeing changes in sex ratio. Not a shocker. This has been seen in lots of other fish species. It turns out if you expose this species to 5 nanograms per liter of EE2, which is an environmentally relevant level, right? This is something you find downstream of wastewater outfalls. You do see a significant difference in sex ratio once you rear those fish up to adulthood. We're not sure what's happening with the F1 or F2 adults yet. That the jury is still out on that, but we are seeing that significantly feminized sex ratio in the fish exposed to ethinyl estradiol. Sometimes effects are a little bit unexpected. So when we looked at larval length, so when we looked at growth, in that exposed generation, that directly exposed generation, we actually saw what almost seemed to be a positive effect of exposure to steroids. So if you expose them to an estrogen or to an androgen or to progesterone, which can act as an androgen in fish, we actually saw that they were significantly longer than the control fish. And then that didn't carry over to the next generation. I guess in a way, it wasn't too unexpected because we know that in humans, if you expose yourself to a steroid, right, you're going to tend to get bigger. So not a shocker. Something that we want to look into, though, is whether there's a trade-off here. You might be getting bigger, but does that mean it's easier for predators to find you if you're larger as a larval fish? We don't know. We also saw deformities, just like we had seen in that other study. We saw, interestingly though, that in the parents, we didn't see any significant differences. But then when you went out to the F1 generation, you did see differences in deformities. And so we saw significant increases in craniofacial, cardiovascular, meaning that the heart was slightly deformed, or skeletal defects, meaning that there was some curvature of the spine, for example. Particularly in the trenbolone and the EE2 treatments in that second generation. Potentially because if you think about it, the F1 generation is getting exposed at a slightly earlier time, right? They're getting exposed when they're primordial germ cells, when they're pre-sperm and pre-egg. So that period is probably more sensitive than the fish would be if they're being directly exposed as embryos, right? And then we also looked at gene expression. I won't spend too much time on this, but looked at some of the same genes as we had in the previous study and saw, not surprisingly, that genes involved in steroidogenesis, receptors, so these are estrogen receptors, as well as receptors in the brain that regulate the production of steroids in the gonads, were altered in some cases. So we still have a lot more work to do at the genomic level here. Lastly, and what's new to this study, is that we really want to get at true, excuse me, losing my voice, epigenetic effects. And to bring it back to this, right, you're only seeing true epigenetic effects, so effects that are not a result of exposure to the stressor in that F2 generation. And so this is work that we're going to be doing in collaboration with the UC Davis Genome Center, which has sequenced the genome of this fish. And the question is, what's happening basically before gene expression gets altered? And what we know now is that gene expression is regulated by something called DNA methylation, meaning that methyl groups get added or removed to the promoter region that is prior to where the actual functional gene exists. So the promoter region in your DNA is basically what decides whether a gene gets turned on or off. And if a methyl group is added to that region, that basically blocks expression of that gene. If it's removed, it increases expression of that gene in general. It's a bit of an oversimplification. And so what we're going to be looking at is which genes are methylated and which genes are demethylated. And what has been shown in other species is that those patterns of DNA methylation can be passed down through multiple generations. Some studies have even shown that this happens through the F5 generation in mice. And so this is the next big question. If we're seeing, once we get to the F2 and looking at phenotypic effects in the F2, can we trace those phenotypic effects back to something that's happening at the DNA level, basically happening epigenetically, which means on top of DNA, the methyl groups being the mechanism. So to sum up, what we're finding some phenotypic level effects. So we're finding changes in sex ratio, changes in growth, right? Changes in deformities, changes in the expression of genes. And now the question is, how are those genes being changed in expression? Are we going to find, when we look at DNA methylation, oh, well, okay, the estrogen receptor gene that was downregulated because the promoter of that gene was methylated, for example. So that's the next bit of work to do here. All right, so moving on to other models, another issue that my group has been thinking about is microplastics. And so lots of concern over this area, lots of studies right now. And really you can consider plastics to be what we call a multiple stressor, right? Because you're not just thinking about the plastics themselves, which if ingested, right, they can get stuck in the stomach, they can cause the organism to absorb fewer nutrients from its food, right? You know, everyone's seen pictures of, you know, charismatic megafauna tangled up in those beer holders, the little plastic rings. But the other issue is that plastics come with kind of a cocktail of pollutants associated with them. And so they're produced with certain additives like bisphenol A or phthalates, and those are used to kind of make the plastics softer or firmer, or whatever they need to be in order to produce the product they're intended to produce. So they can leach some of the chemicals that are used to produce them. And then they're also pretty good at taking on chemicals that are already in the water from other sources. And so plastics have been found to absorb to things like DDT and flame retardants, all the other stuff we put into the water that doesn't mix well with water, tends to like to grab onto plastics. So that's the second issue. So there's the plastic itself, and then there's also the chemicals that might come with it. And so this work stemmed out of a collaboration from UNCW, where I was prior to coming here. And I'd been talking to the director of the microscopy lab there. And she does a lot of work with this really cool organism. It's a protozoan, and it's a tin-tinted ciliate. And I know some of you might be familiar with this type of organism, but they're basically found everywhere, mostly in marine and estuarine environments, but also in freshwater. They're single-celled. And she was doing some work with a PhD student and looking at their feeding behavior. And she was using plastic microspheres to basically take videos of the way they fed and studying how ion channel stimulation was causing them to open up and contract to grab onto their food. And I thought, well, you're using plastics to study this. I wonder if they ingest plastics in the field. And so that got us on to generating a master's thesis out of this to think about how larval fish in particular, who tend to feed on these organisms, might be ingesting plastics that they wouldn't necessarily otherwise see as prey in the field, and how they might be ingesting plastics and how those plastics are trophically transferred from smaller organisms to larger organisms. And so a short video to show you. So this is it's a single-celled eukaryote, and it mostly preys on algae and bacteria. And then in turn, it's preyed upon by copepods and larval fish. And this is a picture of a ciliate ingesting plastic microspheres. So again, this is a single-celled ciliate. It's kind of housed in something called a lorica, which protects it from its external environment. Again, really widely distributed, so on average about 100 of them per liter in marine or estuarine waters. They can be up to several thousand per liter in some cases. And they use their cilia to create a current when they randomly encounter prey. So obviously they can't see or smell or hear their prey, right? But this triggers them to create a current with those cilia, and then the food enters the oral cavity. And they're also able to, in some cases, reject prey. But as far as we can tell, we haven't seen in the videos of these ingesting plastics, we haven't seen that they reject plastics. So they seem, for some reason, to think plastics are perfectly acceptable food, right? So this stimulated us to design a study to look at how larval fish might accumulate plastics from these ciliates, since we know that larval fish ingest them. And so what my student did, she designed a study where she compared ingestion in fish that were fed plastics directly. And these are really small, 10 micron in diameter size plastics, which we know plastics exist in the wild at that size. And so she compared fish ingesting those directly, and she also looked at plastics that were coated with a chemical, in this case DDT, which plastics are known to absorb to, or it's known to absorb to plastics in the wild. And then she looked at fish that were only ingesting those particles from their prey. And what she found was that fish ingested more of the untreated plastics if they were ingesting them from their prey. And if those plastics were treated with DDT, they ingested even more of those particles. What we think is going on there, and that's something that is the next part of the students' work, is that the ciliates themselves might be responding to the DDT and not as able to swim away or get away, contract, when they encounter the fish, when they encounter that predator. And so the fish might actually be ingesting more of the ciliates, or the ciliates might be ingesting more of the particles because their behavior becomes a bit more disorganized because they're responding to that pollutant. But the jury is still out on that. So, right, to sum it up, what we're seeing is that fish are ingesting more plastics from their prey than they do directly from the water. So something to be aware of. And so this work has continued in a study that's been funded by the NOAA Marine Debris Project, NOAA Marine Debris Program. And NOAA's big question right now is, okay, we know plastics are out there. We know that fish are ingesting them. We know that in some cases there are pollutants associated with these plastics. So what does that mean, right? What does that mean, particularly for fish that humans ingest, right? And so that's kind of the next big question is, do these plastics that we're finding across taxa and seafood and marine and aquatic organisms, do they pose a threat to human health? And so we now have a study where we're looking at ingestion in it's an East Coast species of fish known as the black sea bass. And so the question here is, A, are they ingesting plastics in the wild? And so we've already confirmed, this is a case of a fish caught off of North Carolina that has a piece of plastic wrapper. It was kind of wrapped around the contents of its stomach. And then this is another one that, not surprisingly, has ingested a bait fish, right, that fishermen use to attract them. And so now the question is, okay, we're going to do the same study again, but we're going to do this with a commercially valuable fish that's ingested by humans. Are they also accumulating plastics from their prey? And are they more likely to ingest plastics if they're fed them via their prey than if they just eat them directly? And so this is also expanded into some work that's going to start on the West Coast this summer. Obviously not in black sea bass since they aren't on the West Coast, but going to start looking in nearshore rockfish as well. So blue, black, deacon, also potentially cabezon as well, to see if we're seeing the same plastic ingestion in those fish. Thinking is nearshore rockfish are also opportunistic as far as what they eat. They have a similar mouth size, similar distribution of prey, not exactly the same as a species like the black sea bass. And so this work has been funded by the Agricultural Research Foundation to start this summer. So thinking is, leverage some of the methods that we're developing for the NOAA project and bring them here to the West Coast. All right, so the last thing I'm going to talk about today is a model that we've developed to look at the effects of oil spills. This is also relevant to the West Coast in that it uses a fish called the sablefish. And so sablefish, as some of you might know, I know there's some work with this species here from time to time. They're native to the northern Pacific, but they have a huge range. It turns out that you can find them as far south as Baja California and basically all the way to the coast of Japan. There's two subpopulations. So there's a north-south split around Vancouver Island. And so they're barotolerant, even though they tend to be found at depth. They tend to be found at around 400 to 900 meters or so. And they're commercially cultured, it turns out. I found them very tasty. They're also called butterfish. And so they're cultured in Canada, they're cultured in Washington as well. And so we were starting this study a couple of years ago, and we wanted to use a fish that was relevant to deep water drilling, since at that time there had been an increase in offshore drilling. Of course, that kind of tapered off after Deepwater Horizon back in 2010. But really, the Deepwater Horizon spill, as I'm sure most of you remember that, really showed us how little we knew, A, about controlling these offshore rigs if something went wrong. And B, also it revealed how little we knew about the responses of organisms that live at depth. Because we really, most of the studies that have been done on rainbow trout, fathead minnows, things that live in freshwater, at shallow waters, not something that would be experiencing a deep sea release. And so it decided to develop sablefish as a model for responses to these deep sea oil releases. And so they do tend to get pretty gigantic, as you can see there. We were not working with the adults. We were working with small juveniles about this big. Right, and so it turns out that they're pretty sensitive. This wasn't too surprising, considering, if you're comparing something like a sablefish that lives in a clean, semi-controlled environment, right? It's not moving around too much. It's not in an estuary, right, where it's going to be experiencing lots of stressors. It turns out that it's more sensitive to when it's exposed to components of oil, to oil itself, and to dispersant. How many of you remember the whole issue with dispersant use during the Deepwater Horizon spill? That was something we tested separately because, as you might remember, the amount of dispersant used during that spill was just phenomenal and not really thought out until after the fact. And so, again, relatively sensitive compared to other marine organisms. So, for example, expose them to 3 different PAHs. One was toluene, one was 2-methylnaphthalene, and the third was phenanthrene. The idea behind this experiment was to look at a wide distribution of different PAH types, so different sizes, different lipophilicity, or basically inability to mix with water. And so, for example, for 2-methylnaphthalene, which is a typical PAH that's found in crude oil, the LC50, which means the amount of it that was lethal to 50% of the fish, the juveniles that were exposed, was 0.73 milligrams per liter for sablefish. Well, the range in other tested species goes from about 0.35 to 8 milligrams per liter. So they're on the lower end of that range. And so what we wanted to do was get an idea of what sablefish sensitivity is compared to all of the other organisms that have been tested with oil, dispersant, other lipophilic pollutants, so persistent pollutants in general. And so we use something called the target lipid model. So the target lipid model is based on the hypothesis, right, that the concentration for an LC50 in water, right, so something that a fish or a crustacean would represent a response from one of those organisms, that it can be predicted from something called the critical body burden. And so something like 2-methylnaphthalene or phenanthrene is really good at mixing with lipids, mixing with fats. And so if an organism is exposed to a chemical like that, that chemical is going to tend to concentrate in the lipid, right? So if you think about something like, you've probably heard about flame retardants, for example, or certain pesticides like DDT, they get some organisms from the Arctic, like a polar bear, for example, and they find these chemicals in their lipids because they're persistent. They're really good at hanging around in fats. And so that's the kind of chemical that this model predicts the toxicity of. And so what we did is we took all the LC50s from the different polycyclic aromatic hydrocarbons we tested, again, components of oil, and lined them up with this target lipid model that is supposed to predict the toxicity of these chemicals. So down here, you have something called the octanol water partition coefficient. So the higher this gets, that means the more lipophilic that chemical is, the more it likes to partition to the oil rather than partition to the water. And then over here, you have the log LC50, 96 hours. It means, how many organisms were killed over a 4-day period. And so you can see that the results from our experiments with sablefish line up nicely with that model, which means you can use that model to basically predict where they're going to lie on a species sensitivity distribution. Right? And so I have that here. So this is a species sensitivity distribution for many, many different organisms. I think there are about 40 organisms on this chart. And this critical target lipid body burden was predicted using that target lipid model that I showed on the previous slide. And so each of these points represents a value that was generated from lots of different tests. So they were tested against 4 different PAHs or a couple of other different lipophilic chemicals that are pollutants. And then this value was generated as kind of a general measure of their sensitivity. You can see it varies a lot, right? Down here we have an aquatic invertebrate. That's the most sensitive. Up here at the top is algae. Turns out algae is pretty difficult to kill. And then in the middle you have lots of different organisms, from fish to invertebrates to other aquatic insects. And so if you take the results from our testing, looking at responses to PAHs in sablefish, it turns out that sablefish end up right about here. So it turns out that, at least based on the testing that we did, and admittedly, it was only the 3 different compounds, and that should be expanded upon to really get a more solid idea of how sensitive they are, it turns out that this deep water, cold water species that could be potentially vulnerable to oil releases at depth is relatively sensitive compared to other species. And so when you're thinking about risk assessment, often risk assessors will talk about something called the HC5. And the HC5 is a measure of what percentage of animals are affected by a particular chemical. HC5 is a cutoff, and if you're at the HC5, that's representative of 5% of organisms being below the cutoff and 95% of those organisms being above the cutoff, or safe or protected by a particular water quality value, say. And so sablefish are almost a little bit outside that range. And so it does get you thinking about risk assessment, and that is it inclusive enough of organisms that tend to lie at the outer range of these sensitivity distributions. So with that, thinking about future directions, lots of future directions. One of the big questions from the multi-generational studies we've done is, can fish adapt to EDC exposure? Do we see true epigenetic effects, not just at the F2, but would we be able to design studies to go beyond that and think about F3, F4 generation? Thinking also about assessing responses in wild fish rather than just fish that are cultured in the laboratory, for example. Are you seeing different responses depending on geographic location, right, of the same species? I'm thinking about plastic assessment on the West Coast. I'm starting with nearshore recreational fishery species like rockfish, cabezon, and then some projects with potential for West Coast application. Definitely thinking about sablefish and further developing that model, especially since there seems to be development on the horizon of more offshore drilling. Maybe not in Oregon. We'll see what happens, but it's a potential future concern. Thinking about oyster aquaculture, we have another project where we're looking at ecosystem services and the effects of environmental stressors, looking at oysters and whether their ecosystem services that they're providing are affected by stressors in the environment. And then another study relevant to the West Coast is looking at trophic transfer of pesticides. In the San Francisco Bay, they have Hyalella, it's an amphipod that has become resistant to certain pesticides. And those Hyalella are then potentially transferring those chemicals to fish species. So another study we have going on down there. So with that, stop. There we go. Lots of people to thank, both at UNCW and also other locations where I have other collaborators. And I'd be happy to take questions. Yeah.
How are methylated nucleotides passed on to the next generation?
They don't know yet. So we know that they are passed on, but they don't know exactly what the mechanism is. It's known that the epigenome is basically erased and then it is recreated de novo at particular stages within an organism's lifetime. So basically at fertilization, and then it happens again shortly after hatching in fish. And so there's still a lot of questions as to what the mechanism is, but it's known that you see these changes passed on beyond the directly exposed generation.
And is there a selective advantage that people have talked about? I mean, why would you do that as an organism?
There is a selective advantage. Yeah, there is a selective advantage. I'm sure you could think of it as influencing evolution in maybe a faster way. And there are theories that you actually have methylated regions being more likely to undergo mutation. And so the more classical method of evolution may be influenced by DNA methylation, for example, if that makes sense. So that's the new, it's called the unified theory. And then some people are calling it the theory of basically Darwinism and Lamarckian theories of evolution. There's a couple of papers out on that right now, because methylation and epigenetic mechanisms are kind of harken back to Lamarck's theory of how evolution worked versus Darwin's theory. Yeah.
Yeah, regarding your multi-generational, I don't remember, Menidia, are they brooders or are they pelagic spawners? Do they release their eggs into water?
They don't release their eggs into the water. So they release their eggs onto grasses, and then they're
outside the body, though.
That's right.
And then is the water that the eggs, is the water contaminated as well? Are the eggs getting exposed outside the body of the female?
Mm-hmm.
And therefore, it's not the parental exposure, it's the water exposure during embryogenesis.
So for the second experiments that we did, we exposed the parents as eggs. So that exposure was isolated, if that's what you're asking. The exposure was isolated to the parents as embryos and larvae, rather than
multiple generations.
Mm-hmm.
At some point, how do you separate the effects internal to the adult fish, the exposure of the egg, not being the exposure out as opposed to the adult female?
So we didn't isolate that in the first experiment where we looked at the combined effect of climate change and endocrine disrupting compounds, but we did isolate it in the second set of experiments where we didn't expose the parents. We didn't expose adults before they laid the eggs. We only exposed the eggs themselves, if that makes sense.
The gist of my question here is I'm not sure how you're separating the effect of where the exposure is occurring, either in the female or outside the female post, release of eggs.
They exposed the eggs of the F0 generation and then made the water clean.
That's right. So we didn't expose the adult females in that second set of experiments. You're right that for the first set of experiments where we looked at effects of temperature and endocrine disrupting compounds, we didn't isolate that because we exposed the adults and then the adults spawned, and we continued to expose the eggs until 21 days post-hatch. But in the second set of experiments, like he was saying, we only exposed the F0, the parental generation eggs. And so there was no exposure of the adult parents. It was clean water after 21 days post-hatch. Does that make sense? Okay, cool. Yes?
It's kind of a two-parter, I guess. So, being that sablefish are really high lipid species,
Mm-hmm.
do you see that general pattern where maybe like biochemically they have a higher or lower LC50 because of that? And then I guess an interesting question for me, I'm a pinniped biologist.
Mm-hmm.
If lipid-rich species that has more potential to transmit toxins,
Mm-hmm.
but has a lower lethal dose, does that even out over the food chain if they're essentially dying versus? So essentially, would seals or sea lions that have a diet higher in those lipid-rich fish have more exposure, or would that be canceled out because of the LC?
Because they would die anyway. That's a good question. I will say that the levels we were exposing them to in the lab, and that really was an initial set of experiments just to get a ballpark idea of how sensitive they were, those pH levels were higher than what they would encounter in the field. And so I don't know if you can sort of jump to that level yet to predict how it would affect organisms that are consuming them. I think more work would have to be done at sublethal levels to really get an idea of what sablefish accumulate versus what, say, rainbow trout accumulate. But it is a good question, is that target lipid model really relevant to sablefish? Because most of the organisms that we showed on that species sensitivity distribution probably have much lower lipid levels than sablefish do. I guess the other thing to think about is they were only exposed for 4 days, so they probably didn't reach maximum accumulation of those chemicals. You would need to expose them for a longer period of time. But those are both really good questions.
It'll be cool to see with like rockfish.
Mm-hmm. Yeah, that's why I want to do more work with sablefish, basically. Yeah. Yeah.
That was a great seminar.
Thank you.
Appreciated your, especially systems approach coming from gene expression, all whole populations. I had a question about your experiments.
Mm-hmm.
The exposure experiments with the silver sides. Have you done the same thing with populations that are naturally chronically exposed to pollution and so forth versus
Mm-hmm.
those that are
I haven't yet, and that's what I'd like to do. I'd like to actually get subsamples of populations from different parts of the country and populations that have basically evolved to tolerate different stressors. Also, not just evolved to tolerate different stressors, but have evolved at different temperatures. For example, I mean, it's a fish that's distributed basically from Maine to Florida. So how do silver sides in Maine react differently to being exposed to a warmer temperature and a chemical than, compared to fish in Florida, for example? So no, it's a really good question. I haven't, but other studies have shown in other species of fish, like killifish, for example, that fish that have basically been reared in near superfund sites. Right, that you might have read about this study, was exposed to lots of PAHs over many generations. If you take fish from the clean site and move them into that space, you would do a transplant. The fish from the clean site don't make it. It turns out that's because of upregulated levels of certain enzymes, detoxifying enzymes, for example. So no, that would be a really cool experiment. I have plans to propose it. Fingers crossed. Yes.
I have a question about that. First, the transgenerational study of the Christmas stressors. You posed this question about if you're interested in seeing the organisms in adapt. They're just asking for clarification there. So they say adapt adaptation in the extraordinary sense for the scale of population, or do you mean adapt so like
I would say adapt individually. Yeah. So are we seeing fewer vulnerabilities here? If we were able to get funding for a 5-year study, we were able to take it out to say the F5 generation. Do we see deformities in F1, F2, and then start to see a decrease in F3, F4, F5? Are we seeing that phenotype is basically being leaving the generation, if that makes sense, leaving the population. Is that kind of what you're getting at?
Well, that seems likely.
Sure. Okay.
Over time, population generation. I was thinking within the lifespan. You said individualizing within single individual lifespan. So that would be the phenotypicity kind of genetic regulation.
Oh, I see. So if you subsampled a particular set of individuals and looked at DNA methylation throughout their lifetime, are you seeing a change in DNA methylation throughout their lifetime? I hadn't thought about that, but it'd be interesting to look at. So yeah, I guess I was thinking more at the population level.
My second question is the population stuff. You doing that for just that kind of fusion? Is that your estimated
It's a good question. We're planning on doing something called reduced representation by sulfite sequencing, and that would be genome-wide, at least initially. We do have an initial genome sequencing and assembly done on these species, but it's somewhat limited, and so we don't necessarily have enough information about each of those particular genes and the associated promoters to do a gene-specific study. So we're going to start by doing this RDS approach, which basically lets you get at these CPG repeats that are specific to promoter regions. So it uses a restriction enzyme that digests those particular regions, and then you can focus on methylation just in those CPG island regions. So that's what we're going to do first. And then you can kind of back sequence and figure out which genes were methylated, which ones weren't, and then you can kind of get an idea of genes we might target, right, with UPCR, if that makes sense. Yeah.
Once you're in microplastics,
It binds to the plastics. So DDT, we've been with us for a long time. So if you took a sediment core basically in any body of water, you would find DDT a few inches down because it's very hydrophobic and it tends to associate with sediment. In the same way, it's going to associate with plastics, right? DDT can't really mix well with water, so it's going to bind to anything that isn't like water. So it's going to bind to sediment, it's going to bind to plastic. Oh, so you're asking if it's binding to the, okay, I see what you're saying. As far as I know, it's binding to the plastic itself. But it's a good question as to whether a biofilm on the surface of that plastic would increase the ability of, that would increase the ability of DDT to bind. I'm not sure. I don't know that anyone's looked at that. That's a really good question. But regardless of whether the biofilm is there or not, it does have a high affinity for plastic particles. No, it doesn't have to be plastics, but plastics that have been sampled around the world and then solvent extracted. DDT is one of the most frequently detected chemicals associated with it. But yeah, it's a good question as to how biofilms find that out. I don't know that. I don't know that. That's a good question. Yeah, that's a good question. But we didn't see deformities in our controls. So, yeah. Thank you.